Welcome and hi everyone. We want to present our field trip with Practical 1 and Practical 2 in Taman Botani Shah. Our group member is Afandi, Ain, Amirul and Fateh. We almost did our task in the Zingaberales garden. Taman Botani Negara Shah Alam was developed on 24 April 1986, now as Bukit Cerakah Agriculture Park in 1991. The name was changed to Bukit Cahaya Seri Alam Malaysian Agricultural Park, changed to Bukit Cahaya Seri Alam Malaysia Agricultural Park since 2007, has been named Shah Alam National Botanical Garden until now. The objective of the Practical 1 and 2: first, to apply basic statistical method to analyze variation and relationship in leaf length and width data. Second, to use quadrat sampling technique to study the distribution and abundance of plant species in each area. Third, the last one, to related statistical and field data to ecological factors influencing organism distribution and variation. Based on the question number 1 for the practical 1, the mean leaf length differ among the three sets. For the set 2 has the longest leaf with a mean length of 26.2 cm. For the set 1 has a moderate mean length of 21.1 cm, while set 3 has the shortest leaf with a mean of 16.8 cm. This difference shows that leaf length varies between the sets even though the leaves come from the same tree. This variation may be due to factors such as leaf edge position on the tree or sunlight exposure. For the mean leaf width, shows both similarity and difference. Set 2 has the wide leaf with a mean width of 12.7 cm. Set 1 and Set 3 have the same mean width of 7.3 cm. This suggests that leaf width is similar for Set 1 and Set 3 even though their leaf lengths are different. Overall, the comparison, leaf length varies more than leaf width among the three sets. For the set 2 has the largest mean value for both length and width, indicating that its leaves are generally larger. This shows that different leaf sets can vary in size, highlighting the importance of measuring multiple samples to understand biological variation.
By mean, we can calculate the variance and standard deviation. We use the formula the sum of S1 minus S bar square divided to degree of freedom n minus 1 to obtain the variance in square centimeter. By variance show how spread out the data is from the mean. If the variance is high, the leaf measurements are very different from each other. If variance is low, the measurements are more similar and consistent. When we take the square root of the variance, we get the standard deviation. Standard deviation tells us, on average, how far each measurement is from the mean, using the same unit, centimeter. Interpretation for set 1 show that length change more dramatically and more variable than with among the 10 leaf. That because leaf naturally show variation by genetic, sunlight exposure, leaf age, nutrient availability, and position on the branch. Using different set of measurement make the data more accurate and reduce bias. It helps us determine whether the variation is consistent or not.
Assalamualaikum, Dr. Hasya Hannani and all the members involved in my group. Today I will explain about the task has assigned to me. In practical 2, the question is at number 1 and number 2. So the question at number 1 is the term definition of quadrat. So in ecology, geography and biology, a quadrat is a tool used to isolate a standard unit of area or for studying the distribution and abundance of organisms over a larger habitat. Okay, key characteristics. Physical form, traditionally a square or rectangular frame made from materials like wire, wood or PVC, while 0.5 meter times to 0.5 meter is common sizes, vary from 10 cm times 10 cm for mosses to several meters for larger plants. Okay, the purpose is the researchers use them to count organisms within a representative sample to estimate the total population of a larger area. Such stability, they are most effective for sampling sessile, non-moving organisms like plants or very slow moving animals. Okay, we have 3 types of quadrat. The first one is frame quadrat, a simple open square frame used for counting individuals or estimating percentage cover. The second is grid quadrat, subdivided into smaller internal squares to help estimate the percentage cover of species that are hard to count individually, such as grass. The third point is point quadrat, a T-shaped frame with pins dropped through it species if is recorded if it is hit by a pin. Okay. The second question I will answer today is what are the 2 basic requirements of all the quadrat techniques. Okay, the 2 basic requirements for all quadrat sampling techniques to ensure satisfactory and scientifically valid results are: the first one is unbiased random selection. To avoid selection bias and ensure results can be generalized to the entire habitat, quadrats must be placed randomly or through a systematic unbiased process. Every part of the habitat should have an equal chance of being sampled. Adequate sample and quadrat size. The number of samples quadrats must be large enough to minimize the effect of anomalies and properly capture the variability of the organism's distribution. Additionally, the quadrat itself must be an appropriate size relative to the organisms being counted. Furthermore, for these techniques to be effective, the target organisms must be relatively immobile, such as plants or sessile animals, during the counting period to ensure none are missed or counted multiple times. Okay, I'm done with my my explanation and presentation. Thanks to all, to Dr. Hasya Hannani and to all of my friend group member. Thank you.
Next slide, I will answer for the next question. What would be the advantage and disadvantage of using a larger or smaller quadrat to survey the sedentary organism? The answer is using a larger quadrat allows a greater area to be surveyed, which can provide more representative and reliable data for sedentary organism. It reduces the effect of random variation and sampling area. However, larger quadrat require more time and effort to set up and survey, and counting or estimating organism can be more difficult and less precise. Using a small quadrat is quicker and easier to handle and is useful when organisms are very abundant or evenly distributed. However, small quadrat may not accurately represent the whole habitat, leading to higher sampling error and less reliable result, especially if the distribution of organisms is uneven. Next question is, problem with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested area or in aquatic habitat where one cannot directly see the animal or plant of interest. Explain the issues concern ecologists in conducting the sampling method. The answer is, in large forested area or aquatic habitat, spatial scales create several problems for ecologists when estimating average population densities. The habitat is often very large, making it impractical to survey the entire area, so only small samples can be taken. This increases the risk that the samples are not representative of the whole habitat. In addition, organisms are often patchily distributed, meaning some areas have high density while others have very few or none, which can lead to biased estimates if sampling locations are not chosen carefully. In aquatic habitat or dense forests, limited visibility make it difficult to directly observe or count organisms, increasing the chance of underestimation or misidentification. Accessibility is another issue, as some areas may be difficult or dangerous to reach, further restricting where samples can be collected. As a result, ecologists must be rely on indirect method, assumption, or extrapolation, which can reduce the accuracy and reliability of density estimate.
Today I'm going to answer the question number 5. Why there are so many different plant of species in one quadrat, and what are the factors that affecting the distribution? Number 1 is by the abiotic factor. Microhabitat variations. A quadrat is not a uniform environment. Small-scale differences in soil type, moisture, pH, nutrient levels, and light exposure exist even within 5 meter x 5 meter. Number 2 is by competitions and dominance. Biotic factor. Plants compete intensely for space, light, water, and nutrients. More competitive dominant species will form clusters where they win, reducing the numbers of variety other species nearby. These create patchy distributions of different species within the quadrat. Number 3 is by dispersal and chance establishment. Seed rain is not even. Seed falls randomly from parent plants or other dispersed by wind and animals, whereas seed lands is a matter of chance. This leads to an uneven starting point for plant distributions. Some species may simply not have had seeds arrive in particular part of the quadrat. Number 4 is by life history and growth stages. A quadrat captures a snapshot of a dynamic community. It may contain a mix of seedling, sapling, and mature plants of a different species, all growing at different rate and competing for space. Number 5 is by small-scale disturbance. Localized events like animals burrowing, a fallen branch, or trampling can create a small open patch. This resets that tiny area, allowing different fast-growing or pioneer species to establish, adding to the variety within a quadrat. A quadrat is a mosaic of a tiny environmental condition. Each plant species distributions and abundance within that mosaic is determined by the interactions between each species need, that's a niche, and the local conditions plus chance. Therefore, finding a mix of species with a different number is expected and natural results of ecological process working on a small scale. Thank you.